Jeffrey got his bachelor's degree at Northwestern, majoring in chemistry and mathematics. And while he was there, he worked in the laboratory of Richard Silverman, where he synthesized small molecule inhibitors of the enzyme nitric oxide synthase. After graduating from Northwestern, he went to MIT, where he worked in the laboratory of Alice Ting. And he worked on engineering metallo-enzymes to create various types of hybrid probes for cell biology. One of these is a really cool enzymatic tag known as APEX that actually has been used by more than 2,000 researchers around the world. It's actually pretty cool. I wasn't aware of it. I did some reading on it, but it's a way of achieving very highly selective staining of biological systems. And it basically is this enzyme that can be fused onto biomolecules of interest and provide very high selectivity then, and allows electron microscopy techniques to be applied to very delicate biological samples in order to be able to do that. So in 2015, he moved to UC Berkeley as a postdoc. He currently holds one of the very prestigious Miller Fellowships. And when he moved to Berkeley, he also moved programmatically to a very different area, which is more in the area of inorganic materials chemistry. So he's been working in the group of Jeff Long. Jeff Long is one of the leaders in the field of metallo-organic frameworks, or MOFs. And Jeffrey's going to tell us, I understand, mostly about your postdoc work. Is that correct? All right, with that, take it away.
Can everyone hear me in the back? Okay, good. Thank you so much, Bob, for the introduction, and thank you everyone for coming. I'm going to tell you a talk today in two parts, and I will tell you a bit about my work in the group of Alice Ting, but as Bob said, I'll spend the majority of my time telling you about my ongoing postdoctoral work in the group of Jeff Long. And the first part of the talk is on engineered heme peroxidases as genetically encoded tools for cell biology. To motivate this part of the talk, I'll point out that proteins are among the most important molecules in the cell. They execute myriad functions, just a few of which are listed here. And to understand a protein's function, it's really critical to take into account where it is located within the cell. To illustrate this with an explicit example, the protein cytochrome C facilitates electron transfer when it is located within mitochondria, and this is important for respiration. But when that same protein is located in the cytosol, it serves as a signal to initiate cell death. So these are two very different outcomes that can arise depending on the location of this same protein. What we need then is tools that allow us to locate specific proteins of interest within intact cells. But this is quite a challenging problem because our one protein of interest is actually embedded in a very complex and crowded cellular environment. It's surrounded by thousands of other biomolecules, and there is not a unique spectroscopic signature by which our one protein of interest can stand out. An elegant strategy to solve this problem is genetic tagging. Here, we prepare a piece of DNA encoding the protein of interest attached to some tag. We introduce the DNA to cells, and it provides instructions for the cells to synthesize a protein fusion construct. Now the protein of interest is covalently attached to a tag that provides a unique signal standing out above everything else in the cell. The most famous example of this approach is GFP, or green fluorescent protein. This protein is originally derived from jellyfish, and it has the intrinsic ability to glow. So if you attach it to a protein you're interested in, you can now locate that protein within cells using fluorescence microscopy. And this tool has been revolutionary for cell biology because it is robust and easy to use, and it works within living cells. However, there is a serious drawback with this technology, which is the inherently limited spatial resolution that you get from fluorescence microscopy. This is on the order of a few hundred nanometers, which is much larger than most biomolecules that we wish to localize. To illustrate this explicitly, what I'm showing you in this picture is mitochondria stained with green fluorescence. So each of these lines is a mitochondrion, and you can see that it appears to be only a few pixels across. But when we look by electron microscopy, or EM, we get much higher spatial resolution, and now you can see a lot of intricate internal details that were not possible to discern by fluorescence. So EM has the advantage of giving us much better spatial resolution, but it has its own serious drawback, which is that there is no broadly applicable genetic tag for EM. What this means is that if we want a specific protein of interest to stand out within this high-resolution image, there is not a straightforward way to accomplish that. You may be wondering, how would this actually function? If we had a genetic tag for EM, by what principle would it operate? And the scheme I'm going to propose in this talk is shown here. We would attach a tag to the protein of interest, and then we would add cell-permeable reagents, which the tag would convert into a stain in its immediate proximity. And this stain would be dense with heavy elements, which are detectable by electron microscopy. So there is one genetic tag that functions in this general scheme. It is horseradish peroxidase, but it has a critical flaw. This is a 308 amino acid heme enzyme. I'm showing its structure here, and the heme cofactor is shown in blue. This enzyme catalyzes the hydrogen peroxide-dependent oxidation of a wide array of phenols and anilines. It converts them to the corresponding radicals, which are released from the active site. And the reason why HRP is useful for electron microscopy is that it oxidizes this particular substrate, called DAB, into this radical. And this radical rapidly polymerizes to create a highly cross-linked and insoluble polymer that cannot diffuse far from the site where the radicals are produced. Now, this is the key step. We add osmium tetroxide, which can oxidize the polymer, and then osmium remains bound. So osmium is a heavy element that is now detectable by electron microscopy. So we've converted this insoluble polymer into an EM visible stain. So HRP has the advantage of generating this contrast for EM, but its critical problem is that it is inactive in the cytosol and most cellular compartments. This is because it possesses 4 structurally essential disulfide bonds, which I've circled here in the structure. So these bonds fail to form in nearly all cellular compartments, and that compromises activity. And one of the challenges I wanted to address during my PhD was to develop a variant of HRP that does have activity in all cellular compartments. The strategy I used was to look to nature to find enzymes that already catalyzed similar chemistry, but try to repurpose them for my applications of interest. And the enzyme I focused on was ascorbate peroxidase, or APX. This is a 250 amino acid heme enzyme. Again, I'm showing here the heme cofactor in blue. And this enzyme catalyzes in nature the oxidation of ascorbate to the ascorbate radical. And its function is to detoxify plant cells of hydrogen peroxide. For my purposes, the great advantage is that it has no disulfides, so it should function in many cellular compartments. However, there were some challenges that needed to be addressed. The first is that this enzyme is a dimer. This is a serious problem for genetic tags because if you attach something dimeric to the protein of interest, it can cause unwanted aggregation. The second challenge is that this enzyme has not been evolved for activity toward DAB, the substrate I cared about for EM, and you can see that the natural substrate has a very different structure. To address the first challenge of dimerization, I looked at the crystal structure of the dimer interface, which had been solved previously. Here, the 2 subunits of the protein are shown in green and blue, and when we zoom into the dimer interface, you can see a number of electrostatic interactions holding the 2 subunits together. For example, this negatively charged carboxylate is ion paired to this positively charged ammonium. So this suggested to me a strategy of introducing electrostatic repulsion to break apart the 2 subunits. For example, we can mutate this carboxylate to something positively charged. Following this general strategy, I prepared 19 mutants of the protein, isolated them from bacteria, and tested using size exclusion chromatography if they behaved as monomers. And indeed, I found a double mutant which behaved completely as a monomer in solution. To address the second challenge of activity, I used a peroxidase substrate called guaiacol, which generates a convenient colorimetric readout. Unfortunately, I found that APX is about 43 times less active than HRP. This is not promising for highly sensitive imaging applications, and I wanted to understand why is APX so much worse than HRP. To do this, I looked at the crystal structures of the enzymes. Shown here is the active site of HRP, and in yellow is an analog of the substrate. What's striking is that the substrate is surrounded by a cage of phenyl groups, and these phenyl groups have been reported to be important for the activity of HRP. Strikingly, in the corresponding positions, APX possesses polar residues or glycine. So the strategy I wanted to pursue was to graft functional groups from the active site of HRP and transplant them into APX with the hope of boosting its activity towards the substrate I cared about for EM imaging. This strategy was successful. This is the picture we started with, and by progressively introducing HRP-like mutations, I was able to dramatically improve the activity, with the most active mutant being about 28 times more active than the wild-type enzyme. So now I developed some monomerizing mutations as well as activity-boosting mutations, and the next step was to combine them to generate a triple mutant, which is known as APEX. It contains and it is a monomeric protein that has enhanced activity relative to the wild-type enzyme. And in a subsequent study, in collaboration with Stephanie Lamb, my colleague from the Ting group, we performed yeast display evolution of APEX, evaluating 10 million different mutants, and we arrived at what's known as APEX2. This is a single mutant. It possesses only one mutation relative to APEX, and it has greatly improved sensitivity for cellular applications. So I wanted to understand, why was it that just one mutation led to a big improvement in activity relative to APEX? Again, to understand this, we can look at the crystal structures. What I'm showing here is the structure of APEX, and this is position 134 in the protein, which is an alanine in the wild-type enzyme. In APEX2, this is mutated to a proline. And what was very interesting is that when I looked back at the structure of HRP, I found that it already possessed a proline residue at this corresponding position. So it's quite striking that our laboratory evolution of APEX2 arrived at the same solution as nature in the evolution of horseradish. Now the next question is, why is it that this proline residue improves the enzyme activity so much? In a detailed mechanistic study that I don't have time to tell you, I found that this proline residue greatly enhances recognition of the substrate. Remember, this yellow compound is an analog of the substrate. And this enhanced binding of the substrate helps the enzyme to avoid inhibition by hydrogen peroxide. Remember, hydrogen peroxide is a substrate in this reaction. It is necessary for the chemistry that I want to occur, but at high levels, it can also cause unwanted side reactions. So this enhanced binding of the substrate we care about for EM helps to avoid those unwanted side reactions at a key step in the mechanism. So we now have our highly optimized tag, APEX2, and I found that it does indeed oxidize DAB, the substrate of interest for EM. But APEX2 does not only react with DAB, it can also oxidize, for example, a phenol conjugated to biotin. It converts it to a phenoxyl radical, which is highly reactive and can covalently cross-link to endogenous proteins. And this chemistry makes APEX2 useful for spatially resolved proteomics. APEX2 can also oxidize amplex red, a non-fluorescent compound, to generate resirufin, which is a bright red fluorophore. And this chemistry makes APEX2 useful for fluorescence imaging as well as hydrogen peroxide sensing. And we can use almost any phenol or aniline, and it will be recognized as a substrate by APEX2. So depending on which structure we select in terms of the organic small molecule, we can achieve many additional readouts, including chemiluminescence, chromogenic staining, and more. So we have a single genetic tag, APEX2, that can be used for many different applications in cell biology. We have indeed now demonstrated that APEX2 can be used for each of these applications, including electron microscopy, spatially resolved proteomics, and also hydrogen peroxide sensing within living cells. In a separate story that I don't have time to tell you, I developed a split peroxidase reporter, which is useful for detection of protein-protein interactions and also visualization of neurological synapses. I don't have time to tell you all of these stories, but I will tell you a bit about the work we did using APEX2 for electron microscopy. And I'd like to acknowledge up front my collaborators for EM, Tom Deerink and Mark Ellisman from UC San Diego. All of the beautiful EM images I'm going to show you were collected by Tom. The first thing we wanted to do was to target apex to a subcellular compartment with a distinct EM signature, in this case, mitochondria. This cell in the middle contains apex within its mitochondria, and it is surrounded by cells that lack apex. You can see each of these dark objects in the middle is a mitochondrion that is stained by apex. And these mitochondria, which lack apex, are much lighter by comparison. So this gives you an idea of the level of contrast we can achieve, which is very promising. At much higher magnification, we can see a lot of intricate details. As a reminder, mitochondria have two membranes, an inner membrane and an outer membrane. And the innermost subcompartment is known as the matrix. In this experiment, apex was targeted to the matrix subcompartment, and it is not attached to any particular protein. It is floating in that compartment, completely filling it up. And consistent with expectation, dark staining is observed throughout the matrix subcompartment. As a reminder, the procedure for preparing these samples involves staining with heavy metals, and that's what allows us to determine where apex is located. But I'll point out that this heavy metal staining also highlights other structures in the cell, including membranes and ribosomes. And this is useful because it allows us to interpret the location of apex relative to other things in the cell. Another thing you'll note is that the intermembrane space, the other subcompartment of mitochondria, is not stained. That is these light areas on the perimeter and extending inward. And this tells us that apex staining does not cross membranes. This turns out to be quite useful in a number of situations, as I'm about to show you. The next thing we wanted to do was use apex to locate a protein that had not been localized previously. And we focused on a protein called MCU, or the mitochondrial calcium uniporter. It's been known for decades that mitochondria take up calcium, and this plays a number of roles in cell signaling. But it was not known which protein facilitated this calcium transport. In 2011, there were back-to-back Nature papers that agreed on the identity of this calcium transporter as the protein MCU. And the two papers also agreed that MCU is embedded in the mitochondrial inner membrane. But the papers completely disagreed on the transmembrane topology of MCU. One study reported that the N and C termini both face the mitochondrial matrix. The other study reported exactly the opposite. So these are clearly incompatible models for transmembrane topology. And knowing the correct topology has important implications for the binding of MCU to its signaling partners in the cell. We reasoned that apex could be used to resolve this controversy because depending on which model is correct, we would expect very different staining patterns. This is again related to the fact that the staining from apex does not cross membranes. This is what the results look like when apex is attached to the N-terminus of MCU. We can see some dark regions of staining, and these regions are only observed in the mitochondrial matrix. We did not see any of that staining in the intermembrane space. As a quick reminder from the introduction, this is the type of resolution we would get from fluorescence microscopy, where the whole mitochondrion is only a few pixels across. So you can see the level of detail we're getting here is much greater. When we fused apex to the C-terminus of MCU, the same result was observed. Staining was present in the mitochondrial matrix, but not the intermembrane space. An important control was to check mitochondria that lack apex, and in this case, no staining was observed. This confirms that the staining in these upper images was really due to apex. Another important control was to deliberately target apex to the intermembrane space. In this case, we did see staining in that subcompartment, and this tells us that apex has activity there. That's important because it means that the lack of staining in the intermembrane space in these upper images truly means that apex was absent. Collectively, all of these results strongly support this transmembrane topology, where both termini of MCU face the mitochondrial matrix, and a number of subsequent studies have confirmed our finding on this. I don't have time to tell you everything that we've done using these tools for electron microscopy, but I'll just point out that it is now in use by hundreds of labs around the world, and in more than 50 publications, apex and apex-2 have been demonstrated to function in many subcellular compartments, cell types, and also in model organisms. we published a Nature Protocols paper which provides detailed step-by-step guidance on how to use this method. And if any of you are ever interested in using this technique, please email me anytime, because I'm excited to see it in use in the community, and I'm very happy to help. To conclude this part of the talk, I've shown you that engineered heme peroxidases are versatile tools for a number of applications in cell biology, and all of this was enabled by engineering a three-dimensional protein scaffold to recognize a particular substrate that was necessary for these applications in cell biology. I'll move on now to the second part of my talk, which is on metal organic frameworks for cooperative capture of CO2 and enantioselective recognition. I told you in the first part of my talk that I could re-engineer a protein active site to recognize a particular small molecule substrate. But if you look at the scope of guest molecules that proteins can bind to in nature, it is remarkably broad. It is not limited to small molecules, but also includes nucleic acids, other proteins, and even small gas molecules like CO2. Toward the end of my PhD, I became very interested in whether one could harness these binding properties for diverse applications beyond biology, including environmental applications, as well as applications in energy, pharmaceuticals, and more. But a serious challenge is that proteins tend to be quite fragile outside of their host organisms, and this typically precludes their use for addressing these challenges. So I also became interested in the alternative idea of trying to replicate protein active sites in a more robust synthetic system. Specifically, I was quite excited about a class of materials known as metal organic frameworks, or MOFs. These materials possess three-dimensional cavities that are approximately the same dimensions as a protein active site. And it's important to note that these cavities do not exist in isolation. They are part of an extended three-dimensional network where that same pore environment is replicated over a highly ordered structure in all directions. These materials are assembled by mixing together metal ions or clusters with an organic linker, and the metal cluster serves as a node to attach to multiple copies of the organic linker. In this particular example I'm showing you, the metal clusters are here, and the organic linker is here. And depending on which building blocks we select, the pores in these materials can be tuned in terms of their sizes, shapes, and chemical environments, which is quite exciting because we can think about precisely tailoring these pores to bind to a specific guest molecule of interest. And as I already mentioned, that pore environment is replicated indefinitely across the structure. So if we could find just the right pore environment to bind to a particular guest, we would have a very high density of active sites. Finally, in many cases, these materials are robust and they can be easily recycled. So there are many applications that we could think about pursuing with these materials, but what I will tell you about today is two specific problems that I've worked on. And I'll start by telling you about my work on CO2 capture. The motivation for this problem is clear. Emissions of CO2 from burning of fossil fuels have been steadily rising for the last century and have especially skyrocketed in the last few decades. And these emissions of CO2 are driving global warming. Unfortunately, this trend is predicted to continue. Fossil fuels will continue to be burned for several more decades at least. Therefore, there is an urgent need to develop CO2 capture technologies to mitigate these emissions into the atmosphere. It's important to note that about 60% of global CO2 emissions come from large fossil fuel burning power plants. So these are point sources that are responsible for a large chunk of the global emissions, and that makes these sites promising for the implementation of CO2 capture technology. The way these power plants work is to burn fuels that generate power, but at the same time you generate a waste gas stream. What we would like to do is selectively pull CO2 out of that gas stream. And what this then allows is release of nitrogen, oxygen, and water into the atmosphere. And these are not harmful molecules. Now that we've isolated CO2 from the gas stream, what do we do with it? That's a very important question. And there are two choices. One, we could utilize it as a commodity, for example, as a building block in chemical synthesis. The other is that we inject it underground at high pressures for more permanent sequestration. My talk will not focus on this issue, but instead I will focus on the separation step. How do we actually pull CO2 out from that gas stream? And what we need is an adsorbent that has high selectivity for CO2 over these other gases. We would like something with high capacity so that we could use only a small amount of adsorbent and still capture large amounts of CO2. And finally, we need long-term stability, because any adsorbent in this type of process would need to be used many, many times over a long time period. The approach that we've taken in the long group is to create amine-appended metal organic frameworks. The idea is to create a high density of amine functional groups in a three-dimensional cavity and take advantage of the known ability of amines to react covalently with CO2. The way we assemble our material is by mixing together a divalent metal cation, such as magnesium, and this organic linker called DOB-PDC. These building blocks assemble to create a honeycomb-type lattice that consists of hexagon-shaped channels running in the long axis of the material. And each corner of the hexagon is lined with metal sites having this coordination environment, where there are 5 oxygens contributed from the organic linker. And there is a 6th site that we can post-synthetically modify with a diamine. Here, one of the amines coordinates to the metal, tethering the diamine to the framework. And the other amine is dangling in the pore. These materials exhibit some remarkable properties for CO2 capture. What I'm showing you here is a gas adsorption isotherm, where on the y-axis, I'm showing you the amount of CO2 that has been captured, and on the x-axis, I'm showing you the pressure of CO2 to which we've exposed the sample. And you can see that at very low pressures, there's almost no CO2 capture. But you hit a critical threshold where the material abruptly transitions from empty to completely full of CO2. These unusual step-shaped isotherms are indicative of a cooperative mechanism. And what I mean by that is capture of one CO2 molecule in the material makes the capture of subsequent CO2 molecules more favorable. These step-shaped isotherms have some tremendous advantages for efficient CO2 capture in a process. I need to point out at this juncture that any adsorbent we use in such a process needs to be very good at capturing CO2, but it also can't be too hard to get CO2 out. Typically in the process, you would first capture CO2 and then use heat to drive the release of carbon dioxide, thus regenerating your adsorbent and making it ready for another cycle of capture. And this would need to be repeated many times. And if you need a lot of heat to drive the release of CO2, the adsorbent will be too energy intensive to implement in a practical process. So when you have a step-shaped isotherm, it is tremendously advantageous for the capture and efficient release. It is because a massive working capacity can be accessed with only a moderate temperature swing. This is directly related to the ability of these materials to abruptly transition from completely full of CO2 to completely empty. In contrast, traditional adsorbents have a Langmuir isotherm shape, such as these depicted here. And what happens is at very high temperatures, corresponding to this red curve, you still have some CO2 captured in the material. So even with large temperature swings, you can access only a modest working capacity. A lot of work by current and former members of the long group has elucidated the mechanism of cooperative CO2 capture, and I'm depicting that here. What I'm showing at the top is the amine-appended material without any CO2 present. So here are the metal-amine bonds. And upon addition of CO2, it inserts into the metal-amine bond, creating a carbamate with its oxygen coordinated to the metal site. And what was a dangling amine has been converted to an ammonium, and the spacing between metal sites is perfect to allow for highly stabilizing ion pairing in between the ammoniums and the carbamates. So we have chains of ammonium carbamate running along the long axis of the material. And it is that perfect spacing between metal sites that underlies the cooperative mechanism. What happens when we capture the first CO2 molecule is it's not particularly favorable. We generate something charged that is not very stabilized. But capture of the first CO2 molecule makes capture at the neighboring site more favorable, because now we generate something charged that can be stabilized through ion pairing. And this effect is propagated to the third site and the fourth site and so on. And that's how the material can transition so abruptly from empty to completely full of CO2. This model is backed up by single crystal structures. Here's what the material looks like without CO2. There's the metal-amine bond. And here is the structure after CO2 has been captured. Here's the carbamate with its oxygen bound to the metal. At the time when I joined this project, there were two main challenges we needed to address. The first is we needed to tune the adsorption properties to make it usable for different CO2 capture applications. And the second is that we needed to improve the long-term stability of these materials. The motivation for tuning the adsorption properties is that there are many real-world CO2 capture applications, and the partial pressure of CO2 in the relevant gas streams can vary widely. It can be very high pressures, ranging all the way to very dilute pressures. And what you could envision is if we could change something about the structure of our material to tune the thermodynamics of CO2 capture, we would expect that the position of this CO2 adsorption step could be moved. And we could think about precisely tailoring these adsorption properties to match the conditions of a particular process to optimize the energy efficiency. I was very excited about working on this because what we had available to us is the ability to change the diamine structure within our material. We had available a convenient post-synthetic modification strategy where we could test hundreds of commercially available compounds. And this changing of a functional group in a three-dimensional cavity was very reminiscent to me of what I had done during my PhD, where I mutated amino acids in a protein active site. The specific changes we wanted to make were the following. First, you could think about introducing a bulky group on this metal-bound amine. If we do this, it should weaken the metal-amine bond, thus destabilizing the starting material of this reaction. This should make CO2 capture easier to occur. Alternatively, we could introduce a bulky group on this dangling ammonium. This should destabilize the product of the reaction, which is chains of ammonium carbamate. And this should make it more difficult for CO2 capture to occur. So these two changes to the diamine structure should have opposite impacts on the thermodynamics of CO2 capture. And following these basic strategies, we have now tested over 70 diamines in the context of this framework. This is work done in collaboration with Rebecca Siegelman and Phil Milner, colleagues in the Long Group. And we found that we were successful in tuning the adsorption step over nearly 5 orders of magnitude. So we did indeed come up with a class of materials that was highly tunable in terms of the adsorption properties. I mentioned that one thing we did to alter the thermodynamics was to weaken the metal-amine bond. But in the course of the experiments, I found that this created a serious problem. When we heat up the sample, which is necessary to release CO2, I found that the diamine was prone to volatilization. It would transition to the vapor phase and leave the material permanently. This leaves behind vacant metal sites, and it corresponds to reduced CO2 adsorption capacity because we rely on those amines for capture of CO2. What this corresponds to is poor long-term stability in these materials, and we needed to improve on that. The strategy we used to improve the stability was to use diamines with this type of structure. On one end of the diamine, we have a primary amine, and on the other, we have a secondary, where we can vary the identity of this R group. We found from crystal structures that this diamine coordinates to the framework exclusively through the primary amine. And what we've hoped is that by using bulky alkyl groups in this position and using a higher molecular weight compound, we could minimize the volatilization and improve long-term stability. And indeed, this worked. Going from a small ethyl group up to a large N-hexyl, we did indeed limit that volatilization. Now, we need to ask ourselves, by introducing something bulky in this position, did we impact the CO2 capture? Before I show you those results, I need to introduce a slightly different assay, which I'll be showing for the rest of the talk. In this experiment, we expose the sample to a constant pressure of CO2. We start at a high temperature, and we slowly cool. Here, we can still see those sharp adsorption steps, and we get essentially the same information as from the gas sorption isotherms I showed earlier. This is just a more convenient experiment. And what we found is that when we used the small ethyl group, as expected, there was a single adsorption step. But surprisingly, when we went to any alkyl group larger than ethyl, now we unexpectedly saw 2 adsorption steps. In each case here, we see one step at 120 degrees, accounting for half of the expected capture, and then the remaining expected CO2 capture was accounted for in a subsequent step. And the spacing between these adsorption steps got larger and larger as we increased the size of the alkyl group. This 2-step behavior is undesirable. I mentioned that the advantage of these materials is accessing a massive working capacity in a single sharp adsorption step. And here, we are effectively cutting that capacity in half. I wanted to understand why is this happening, and if possible, can we devise a solution? To do this, I looked at the crystal structure of the material after CO2 had been captured. Here, this is with the diamine containing the ethyl group, and in this hexagon-shaped channel, each corner corresponds to a chain of ammonium carbamates. From this perspective, the chains are running into the screen. And what you'll notice here is that this channel is not a perfect hexagon shape. The chains of ammonium carbamates tend to point towards each other in pairs. And you can see that this ethyl group, the 2 ethyl groups from neighboring sites are very close to each other. This different perspective of the same crystal structure shows it even more clearly, where these ethyl groups are essentially making a van der Waals contact. What I hypothesized is that when we went to anything larger than ethyl, we were creating steric interference between sites. And this steric interference was preventing the full capture of CO2 at every site in the material. And if this hypothesis is correct, what I needed to do is alleviate steric crowding at this key position. The approach I took to address this problem was to use an isomeric framework. Shown here is the framework I've been showing all along, and this is the organic linker that we used to build it. It has phenoxides in opposing para positions. The linker used to assemble the isomeric framework is very similar, except it has carboxylates in opposing para positions. And what you'll notice is that the isomeric framework has some very similar properties. It has the same molecular weight and an identical pore diameter, but there is a subtle difference in the geometry. It may be hard to see from these structures where solvent molecules are bound to the metal sites, but these arrows should help you to see it. In the parent framework, the metal sites point towards each other in pairs. But in the isomeric framework, there's a more symmetric distribution of the metal sites. So my hypothesis was that this could alleviate that steric crowding. And indeed, this was successful. Now, using even the largest dangling alkyl group, as big as an N-heptyl, yielded a single adsorption step. So we've now generated materials that have improved stability by using a higher molecular weight diamine and maintained a high working capacity. So now that we've addressed these 2 challenges of making a tunable class of adsorbents and improving their stability, in ongoing work we are scaling up these materials and validating their properties for specific CO2 capture processes. I'll move on to tell you about my work using MOFs for enantioselective recognition. I mentioned that proteins are remarkable in the scope of the guest molecules they can bind. But one of the most striking things about proteins is that they often bind individual enantiomers of chiral compounds with perfect selectivity. This is because of the fact that the protein itself is a chiral molecule, and it has an active site that is a 3-dimensional cavity having a chiral shape. Because of this, and because many pharmaceuticals bind to proteins for their activity, opposite enantiomers of chiral pharmaceuticals can have dramatically different effects. These are just 2 well-known examples where one enantiomer of the compound has beneficial therapeutic properties, but the opposite enantiomer has grave side effects. Because of this, it's imperative to isolate and evaluate individual enantiomers of drugs, and this issue is quite widespread because more than half of pharmaceuticals are chiral compounds. Therefore, methods to isolate enantiopure compounds are really critical in the development of new pharmaceuticals. These are 2 general strategies by which you can isolate enantiopure compounds. In one case, you could do a chiral separation, where you start with a mixture of 2 enantiomers and use a chiral adsorbent to selectively pull out one that you're interested in. Alternatively, you can do enantioselective synthesis, where you start with something achiral and you use an enantioselective catalyst to synthesize just one enantiomer. Regardless of which strategy you select, what's needed is a chiral host that binds enantioselectively either to an adsorbate in the case of a chiral separation, or a transition state in the case of enantioselective synthesis. And chiral MOFs are quite interesting targets to utilize as hosts for these applications. This is because of the fact that they have these tunable three-dimensional cavities. And indeed, there are some successful examples in the literature where chiral MOFs have been used either for chiral separations or enantioselective synthesis. I'm just showing two examples here, along with the organic building blocks that went into the materials. So although a lot of work has been done with chiral MOFs, there are some common challenges that are encountered in these materials. And to the best of my knowledge, no published chiral MOF simultaneously overcomes all of these disadvantages. The first common issue is that these materials are often synthesized from expensive starting materials, which means that they can't be made on a practical scale. It's also common that the pore diameters are small in these chiral MOFs, and pharmaceuticals tend to be quite large, which means they can't even fit into the pores. Finally, a lot of these materials have poor stability to air, water, or high temperatures. The MOF that I was working with during the CO2 capture project, I realized was actually a chiral MOF. This material, magnesium DOB-PDC, is chiral because of the fact that the organic linker is permanently locked into a twisted conformation that is chiral. Also, each corner of the hexagon-shaped channel is a metal oxygen helix. From this perspective, the helix is running into the screen, but here I've laid it out from bottom to top. So you can see if you trace the oxygens and the metal sites, it is a left-handed helix, and this is a chiral structure. In this structure I'm showing here, each corner is a left-handed helix. There are no right-handed helices. And I was quite excited about this realization because this MOF has some advantageous properties for chiral separations. It's made from cheap achiral starting materials. It can be made on large scale and turned into pellets. It has large pores on the order of 1.8 nanometers. And it is stable up to 400 degrees Celsius. And perhaps most importantly, as I showed in the first part of this section, amines can be grafted post-synthetically to this material. So we can think about precisely tailoring this chiral three-dimensional cavity to bind to specific guest molecules of interest. So although this material was quite promising, there was a serious issue, which is that this material is actually racemic in bulk. It has an equal propensity to form a left-handed structure and a right-handed structure. And any material that is racemic in bulk will not be useful for a chiral separation. So this is the situation we have. We have these 8 chiral building blocks, and we're making an equal amount of left and right-handed MOF. But what I hoped I could do is add a chiral spectator during the synthesis to bind to the framework in the early stages of its growth and induce it to grow preferentially as one enantiomer. Before attempting this, I had to think about how could I even characterize the material to determine if I had made something that was non-racemic. There are a few methods in the literature for this. One is solid-state circular dichroism. However, this method is not quantitative unless you have an Enantiopure standard of your exact material of interest, and we did not have that. Another approach is single crystal X-ray diffraction. However, we did not have sufficiently large single crystals for this to work. So the established techniques were not going to be suitable, and I needed to develop a new assay to characterize the enantiopurity of my material. So I tried to think unconventionally to develop a new approach to think of the most simple way that we could tackle this problem. This is the issue I was facing. We have 2 MOF enantiomers that are very hard to distinguish from each other. But I realized that if I grafted an enantiopure diamine, I could now convert those enantiomers to diastereomers, which should differ in some physical property. And there are a number of ways that you could think about distinguishing these 2 diastereomers, but the approach I ended up using was to probe the CO2 capture. So I already told you that when you append diamines to this framework, it can exhibit sharp adsorption steps of CO2 capture. And because these are diastereomers, you can envision that the thermodynamics of CO2 capture may be subtly different. So you may actually see two adsorption steps corresponding to the two diastereomers. And this is how the data look. When the Enantiopure diamine is appended to the racemic MOF, indeed, there are two transitions. And I have confirmed that these correspond to the two diastereomers of the material. Now what you could envision, if we succeed in making a MOF that is non-racemic, the data should look like this, where one of the two steps is much taller than the other. And this should be a quantitative approach. So with this straightforward assay in hand, I proceeded to test a number of chiral spectators. These are cheap, commercially available compounds, including a number of amino acids, as well as other natural products. The workflow is as follows. We assemble the MOF in the presence of the chiral spectator, and it's important to note that upon synthesis, the chiral spectator might remain bound to the framework. So I wash extensively to remove any spectator that might be present. This should leave behind just the MOF, which we hope is enantio-enriched. Only now, after all of this has been done, do I introduce the Enantiopure diamine and assess CO2 capture to characterize whether I made something enantio-enriched. So using this workflow, I tested each of these spectators, and the data of the assay are now presented as a single number on the y-axis, where 50% corresponds to the racemic material, which I've shown in gray for orientation. In this experiment, 3 equivalents of spectator were added in each case. And what's quite promising that you can see is many of the spectators indeed induced the MOF to grow preferentially as one enantiomer. But what's not promising is that these numbers are not very close to 100%. We wanted to get closer to 100% enantiopurity. So for further optimization, we selected this chiral spectator. And the reason we picked this one is in data I don't have time to show you, I found that this spectator produced the highest quality MOF in terms of crystallinity and porosity. This is the compound called panthenol that I used for further optimization. It is a chiral alcohol that is an active ingredient in moisturizers, lotions, and shampoos. So it's industrially produced, it's non-toxic, and it's quite cheap, which is important because if we were to use this in the synthesis of a chiral material, it would not greatly increase the cost. I found that by increasing the amount of panthenol in the reaction and tweaking the reagent concentrations, I could succeed in creating a MOF preparation that was 95% enantiopure. And importantly, I found that storing this material in methanol for 10 months did not impact the enantiopurity, which is important because it indicates good stability in this protic solvent. In data I don't have time to show you, we found that this MOF was the right-handed enantiomer, which means the structure that contains right-handed helices. And separate alternative synthetic conditions yielded the opposite enantiomer with greater than 90% enantiopurity. So I don't have time to show you all of these experiments, but I'll show you one simple control. Here in green is the data I've been showing all along with the right-handed MOF and the right-handed diamine. But we would anticipate that if we simply used the left-handed diamine, now instead of a single step at 160 degrees, we should see one step at 120 degrees, and that's exactly what we observed. So this simple experiment provides some compelling evidence that we indeed succeeded in making a MOF that was enantio-enriched. What you may have been thinking about all along is that I've shown that the two adsorption steps corresponding to these diastereomers are in quite different positions. This corresponds to an enthalpy difference of about 20 kilojoules per mole. I wanted to understand, at the atomic level, what is the structural basis for this enantioselectivity? And can we take advantage of that for other types of chiral separations? I succeeded in determining high confidence structures at the atomic level for these two diastereomers. This is with the diamine enantiomers bound and CO2 captured. This was done in collaboration with many colleagues in the long group, combining X-ray diffraction, solid-state NMR, and density functional theory. I don't have time to tell you how we combined all of these techniques to arrive at these structures, but I'll just walk you through the key features of the structures. There are many similarities between them, but also some key differences which underlie the enantioselectivity. The first observation is that in both diastereomers, chains of ammonium carbamate were observed in the long axis of the material, and this was not a surprise because other diamine variants had exhibited similar chains. What was surprising is that there was also an ion pairing interaction between chains of ammonium carbamate, and that's what I'm showing here. We had not observed this in other variants of the material, and what's quite interesting is that this interaction was enabled by the framework distorting its shape. Shown here is how the framework normally looks, where it's closer to a perfect hexagon. Here you can see it's pinching inward substantially, and this is enabled by the organic linker twisting and also bending slightly. So it's quite interesting that the MOF can change its shape to enable strong interactions between bound guest molecules. This is now a different view of that structure. Here are the ion pairing interactions in the long chains. Here is the ion pairing between chains of ammonium carbamate. And both diastereomers show these key ion pairing interactions, but you can see in this diastereomer, the distances are substantially shorter, suggesting that this diastereomer should be more stable, and that is indeed consistent with the CO2 capture results. This is now a space-filling model of the same structure. As a reminder, the diamine contains a cyclohexane ring. Each of these objects here is a cyclohexane. And what I found is that this diastereomer had more favorable van der Waals interactions. It may be hard to see the subtle difference between these two, but these arrows should make it more clear. I found in this case, there was more favorable van der Waals contacts between cyclohexanes and also for the cyclohexane interacting with the wall of the MOF pore. In this diastereomer, this angle leads to less favorable van der Waals contacts. So again, this is suggesting that this diastereomer is more stable. So overall, there are a large number of non-covalent interactions leading to a high degree of enantioselectivity. And the role of the MOF is to interact with the bound guest molecules, but also to serve as a chiral three-dimensional channel to suitably orient the guest molecules to interact with each other. Now I've shown you that in this material, we have all these advantageous properties, but additionally, we now have it in an enantiopure form, and it exhibits a high degree of enantioselective recognition, at least for the case we've characterized in detail. And in ongoing work, I'm exploring the use of this material for chiral separations and enantioselective synthesis. To conclude my talk, I've told you two stories in quite different areas of chemistry. But the overarching theme between these two areas has been the engineering of three-dimensional scaffolds to optimize their function for a particular application, whether it's biological or environmental. And for my proposed work, I'm going to continue working under this broad theme with the goal of creating cavities that have a higher degree of chemical complexity, with the goal of replicating some of the catalysis that occurs in natural enzymes. I'd like to acknowledge a number of people. First, my advisors, Jeff Long, Alice Ting, and Rick Silverman, whose work I did not have time to talk about. They've all been indispensable in my development as a scientist and very supportive of my career, so I'm thankful to them. I benefited from having fantastic colleagues both while I was at Berkeley and MIT, and they were a really important part of the work that I showed you today. And I also had excellent external collaborators both while I was at MIT and Berkeley. And I did not have time to show you most of this work, but they were nonetheless an important part of the story. I'd like to thank these agencies for supporting me, and these for supporting the research. I'd like to thank you for your attention, and I'm happy to take questions.
Let's try to take some questions from the students first.
When you were engineering the active side of the apex, were you expecting to get the promiscuity that you're going to end up getting at the end, where you had a variety of different substrates?
Yeah, so I skipped over that part, but the enzyme whose activity we were striving to replicate, HRP, is also very promiscuous. It's because that active site is very solvent exposed, so it recognizes the phenol or aniline, but then regardless of what's attached sort of on the back, it doesn't matter because it's in the solvent. And so our hope was that we would maintain that promiscuity because then we could do all those different things, and essentially it has a similar substrate profile.
Two quick questions. Firstly, MOFs are known to have oftentimes limited windows of pH stability. Do you know whether your CO2 capture MOFs are stable to things like NOx or SOx and the other various pollutants you get that can oftentimes cause extreme environments in the MOF?
Yeah, that's a great question. So I did not talk much about our validation for the MOF in the presence of a lot of the gas molecules that you would encounter in the process. So oxygen is fine, we've confirmed that. Water, when we use the right diamine, is okay. Well, the problem you can have is that water can replace the diamine on the metal site. But by using the correct, strategically designed diamine, we can avoid that problem. Now, SOx and NOx are more challenging for us to characterize safely in the lab. So we just are getting set up to do those experiments. And so far, it's looking pretty promising, but we haven't fully characterized that. But indeed, you're right, those are very important for us to look at for the real process.
And then a second very closely related question. For your CO2 capture MOFs, do they primarily deposit in monolayers? I know there's some chemabsorption going on there. Do you have multiple stacking layers?
You're talking about the captured CO2 or the MOF itself?
Captured CO2 in the form.
Stack in monolayers.
Monolayers?
Well, so the mechanism of CO2 capture is a covalent reaction where it inserts into the metal-amine bond, and it's these ammonium carbamates. So for every diamine, there's one CO2 that gets captured.
Okay.
Now, at high pressures, it's conceivable that extra molecules could be captured. And indeed, actually, we see that when we go to very high pressures. But for most of the applications I demonstrated, it would be pretty much one CO2 per diamine.
I'm curious about how well the molecules work at point sources, like at the concentrations of CO2 you find at point sources, and also just the ambient atmosphere, and also about the reversibility cycle, how we unload and reload.
Yes. So the reason why we wanted to tailor it was so that we could use a different adsorbent for each situation. So we indeed have some that we could put on the bench right here, and it would fill up with CO2. And those ones can also capture CO2 from the flue gas stream, because CO2 is actually more concentrated in that case. But the reason why we use different variants is the energy required to release. So we just use whatever material can do the job for a particular gas stream and minimize the energy required to release it. So in cases when it captures it from air, that's a case where the enthalpy is very high and we need more heat to release it, but we can't get around that because of the thermodynamics. And now the stability, it depends on the diamine. We have validated some of our variants over 2,000 cycles of capture and release, and we see no loss in performance. So indeed, it's looking quite promising, and we're moving to larger scale in those cases.
The releases with temperature, heat?
Well, you can also use vacuum or a combination of temperature and vacuum, but the most common is to just heat the sample.
Yeah.
So I have a question about how the CO2 moves into the material. Do you think that when these carbamates form, is there a reversibility so that you can form at one site and then pop off and then move to another? Because the question is, how do you fill the whole thing?
Yes. Certainly we have evidence that once you capture it, it's going to be a reversible process. Even if it's not, though, it makes sense that you could fill every site. And the reason is that there's still room in the channel. So we have about 1.8 nanometers, and it gets a little more bulky upon CO2 capture, but still plenty of room for diffusion. And I've done a detailed kinetic study that we haven't published yet. And I found that diffusion was not even the limiting factor. It was the chemistry that was happening.
Okay, and then a somewhat related question. So in principle, if you went from biphenyl derivatives to terphenyls to tetraphenyl, do you just widen out the pores, or at some point do these things start to interpenetrate?
That's a great point. So actually in 2012, Omar Yaghi had a paper where he did 2, 3, 4, 6, all the way up to 10 actually. And he showed that he could prepare those structures without interpenetration. However, it's not clear how good the crystallinity was. I think that it gets very hard to make a highly crystalline material when you're using these massive organic linkers. And actually one other side point is we did explore the use of the 3-phenyl ring linker with these diamines. And it's also, now this is getting a bit detailed, but if you remember, I had this problem of steric crowding, and we also solved that problem by going to the bigger MOF.
I'm curious about some of the inorganic chemistry behind the changing in the ligand from an amine to essentially a CO bond for your magnesium center. Does that change the bond lengths for the surrounding magnesium-oxygen bonds? Do you see any breathing in the structure, whether it shrinks in size or expands respectively?
Yeah, so I would have to go back and check the exact bond lengths, but my recollection is there's not any substantial change in the environment there of the magnesium-oxygen bonds in the framework. There's another paper that's been studied, another paper that has been published from the Long group, where it was the iron derivative of this framework, and it was carbon monoxide capture on the metal site. And actually, there was a change in spin state, and there was a really big change in those oxygen-iron bonds. But in our case, it was pretty much exactly the same.
So I have a quick question. So in the energy storage area, people talk about the volumetric energy density and basically the specific energy density of energy stored in 3 different ways, important figures of merit, right? So if you look at what you're doing in this field, are these also important figures of merit, and how does this compare with other alternative strategies that people are looking at?
That's a great point. So indeed, the amount of CO2 that you capture, the capacity, is very important. And one of the things I emphasized is also the working capacity. So with a given temperature swing, what's the difference between how much you capture and how much can get released? So we are doing really well in terms of the working capacity. It's on the about 18 grams of CO2 for every 100 grams of the material. And the most mature technology for CO2 capture is aqueous amine solutions. So you would think if you just had a diamine as a liquid, that would have a very high density. But the problem is that those amines in pure form are very corrosive to the plant infrastructure. So they have to dilute it a lot with water. And it ends up actually only having a working capacity of about 2 grams per 100 gram, much, much lower than our MOFs. But, and I'll just point out that now the disadvantage we have is that heat transfer in our solid is worse than those liquids. So it remains to be seen once we go to a larger scale and the engineers work on it. It's not clear if we're actually going to beat them, but it has, in terms of working capacity, it's outstanding. Yes.
Just 1 or 2 more, and then we'll have to break.
I guess this is a related question, but imagine you have your MOF that's captured CO2. If you were to heat the MOF using like combustion of gasoline, relative, like how much did you just release versus how much did you burn? Like is it actually efficient?
Yeah, that's the key questions. And that's why nobody wants to implement carbon capture technology right now. So the most mature technology, which is those aqueous amine solutions, gives you about a 35% energy penalty, which means that 35% of the energy from your power plant is used just to run that process of capture and release. And we really would like to get down to about 10%. And with our MOF, if you just look at the basic properties that we've characterized in the laboratory, we actually could get somewhere in that range, but it's not validated yet in terms of a large-scale process, and heat transfer is going to be a really critical issue.
For enantiomeric separations, you'd really like to have Enantiopure MOFs, and you get pretty high. An alternative would be to use a linker that's Enantiopure. I don't remember the exact structure of the DOB-PDC, but can that or some simple variant of it be made as the Enantiopure linker?
Yes, that's one strategy that's been used in the literature. If your linker is chiral to begin with, then you're guaranteed to make a chiral material. The ones that have been published, though, are more expensive than the simple starting material I used. And yeah, it's just going to be a balance of, the one that I showed you, which has 2 phenyl rings, actually is not so amenable to additional groups being attached. They're based on the structure, it will kind of disrupt framework assembly. If you go to the one that has 3 fused phenyl rings, we can put groups on the central phenyl ring. And indeed, yeah, that would be quite feasible, and you'd be guaranteed to make an Enantiopure MOF, but it's a question of the cost of synthesizing that larger compound.
Last question from our department chair.
So I'm struck by the analogy between what you're trying to do and the problem of oxygen delivery by hemoglobin, where in that case the binding and release is a function of the oxygen concentration, but also chemical determinants like pH. And I wonder if there's any mechanisms to use internal allosteric or external control of some other chemical property to facilitate the release so that you don't need to use heat the same way.
Yeah, that's a great point. The only thing we've done that I think might count as an example of that is in some cases, the thermodynamics differ depending on whether water is present in the gas stream. Perhaps our hypothesis is that there's additional hydrogen bonds that stabilize these structures. And so I think it's feasible there could be some kind of a chemical trigger that would destabilize and make the release more facile. I guess it would need to be something very cheap and easy to implement though. But yeah, that's certainly worth thinking about more. In terms of how hemoglobin has this elegant allostery, we don't have nearly as complex a structure, so we're going to be a bit limited in terms of having some kind of responsiveness to various analytes. It needs to be kind of a very direct binding onto the chains.
But you already have cooperative binding, which
Yeah.
is intrinsically analogous.
Okay, I think in light of the time, we're going to have to move on. Let's thank Jeff for a great